Hello everyone, I am Purvi Bhise from B Farm 5th SEM, student of PR Patil Institute of Pharmacy, Talegaon. I am thrilled to be presenting on the topic pharmacognostic study of belladonna. So let's get started. Content of my presentation are introduction, synonyms, biological source, geographical source, cultivation and collection, microscopic character, chemical constituent, adulterant, isolation, uses and marketed products. Now, introduction. Belladonna or deadly nightshades, Atropa belladonna is highly toxic plant native to Europe and parts of Asia, known for its dark purple flower as shown in the picture, blackberries and use of its potent alkaloids for medicinal purpose and historical poisoning. The plant's active compounds including atropine, hyoscyamin and scopolamine acts as acetylcholine antagonists, relaxing smooth muscles and dilating pupils, a practice histologically used to make eyes appear more seductive according to the International Journal of Homeopathic Science. These are some pictures of belladonna. Synonyms: deadly nightshade, the most common name, belladonna herbs, belladonna leaves, or folia belladonna. Friendwort, dwell dates herb, dweberry, and poison black cherry. Family, Solens family. Biological source: the commercial drug is derived from the dried leaves and root of Atropa belladonna. Geographical source: native to Europe and found in the western Himalayas. Cultivation and collection. Cultivation, collection and preparation. Belladonna is grown from seeds. The leaves are said to be richest in alkaloids at the end of June or in July, and a sunny position is said to give more active leaves than a shady one. Plants about 3-year-old are significantly large to give a good field of leaves, and if roots are being collected, it could seed to be best to replant about every third year. 2 or more crops of leaves may be collected annually. Leaves left in an imperfectly dry state dehydrated and give off ammonia. They should therefore be dried immediately after collection and be carefully stored. Leaves of good color may be obtained by drying in thin layer starting with a moderately heat which is gradually increased to about 60 C and then gradually decreased. Sometimes the leaves are badly attacked by insects and roots by fungus. It requires altitude of 1400 meters from the sea level. Now, microscopic characters. Leaves type simple, form broadly ovate, color yellowish green, arrangement alternate, arranged in pair on the upper stem, each pair with large and small leaf, margin accumulated, surface slightly hairy, petiole 4 cm length. Flower color is purple, fruit color green to dark purplish black, type berry. Order, slight and characteristics. Taste bitter and acrid. Microscopic character. Dorsventral leaf, epidermal cells with striated cuticle layer, anisotytic stomata, collenchyma above and below the midrib, unicellular covering, trichomes and unicellular glandular trichomes, calcium oxalate crystals. Chemical constituents are: belladonna herbs contain 0.3 to 0.6% of alkaloids. The chief constituent is hyoscyamin. The other alkaloids are atropine, apoatropine, and trace of hyoscine. Small quantities of volatile bases such as pyridine and N-methylpyrrolene are present. The leaves also contain a fluorescent substance beta-methyl absence. They yield about 14% of ash and not more than 4% of acid insoluble ash. These are the structures of atropine, tropic acid and cinchona. Litolaca candra lineates. Lamina denser, less decurrent than belladonna. Epidermal cell have the straight wall with needle-shaped crystal of calcium oxalate and stomata is of anomano. Let's see how atropine is isolated from belladonna. For isolation, we required belladonna leaves, then clean it, dry it, and powder it. Then treat it with sodium carbonate solution to make alkaloids base free. Then, to utilizing as solvent, ether or benzene is added. By utilizing a solvent by Soxet apparatus, till continuation free volume of solvent is added into that one. Extraction is continued. A colorless liquid may obtain from cyclone tube. After that one, from the extracted that collected in beaker, that is extracted treated with acidifier water. It is shipped in a separating funnel. So separated this all as ether. Ether is separated from the water. So ether remains on top and acidifier water is remain at the bottom side. Now collected, so in acidifier water the alkaloids are present. Utilizing the water for the process, in that case we have to add ether until the acidifier water to removing the coloring matter. Then again separate by utilizing separating funnel, then to add in acidifier water sodium carbonate, it get precipitate out. Filtrate it and wash. Dry this one. Whatever the dried mass are obtained on the filter paper, which we dissolve in water with solvent ether again, and add anhydrous sodium sulfate. After filtration we get crystal concentrated this and cool the filtrate, get crude crystal of hyoscyamin atropine. Add alcohol and NaOH in the solution. On standing hyoscyamin racemize to atropine, we get crude atropine optically inactive, in which add acetone evaporates and we get atropine, a pure atropine. Now, uses of belladonna as local anesthetic and anodyne externally to relieve pain, used to check profuse perspiration of patients suffering from tuberculosis. Atropine dilate the pupils and is used by ophthalmologists. Internally used as sedative and to check secretion. It used in as a plaster in knee joint, elbow joint, lumbar joint as shown in the picture. Belladonna leaves are used in the tincture preparation. It is also used as an antidote in opium and chloral hydrate poisoning. Belladonna are given for common cold, fever, asthma and Parkinson's disease and many others. These are some marketed products of belladonna such as tablets, ointment, homeopathic medicines. I express my heartfelt gratitude to Dr. Nilesh Banarasi sir, our esteemed guide, for his continuous support, motivation and valuable guidance. I am thankful to respected principal sir, director sir, all the faculty members and non-teaching staff for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral supports. Thank you.